Hello, my name is Sophia Honeycutt. I'll be presenting my lab midterm topic. If you had a compound microscope, what would you like to take a closer look at? First on the list are the adorable tardigrades, also known as a water bear or a moss piglet. Tardigrades are a phylum of invertebrates, under 1.2 millimeters in length, which makes it hard to fathom that they are still considered an animal. Tardigrades are capable of surviving temperatures as low as negative 200 degrees Celsius, temperatures as high as 151 degrees Celsius, changes in environmental tonicity, extreme levels of high pressure, extreme levels of low pressure, and amazingly, extreme levels of radiation. Due to their resilience, tardigrades are even able to survive the vacuum and solar radiation of outer space. Next up are cancer cells versus healthy cells. Under the microscope, cancer cells would appear to be mutated, abnormal in growth rate, shape, size, arrangement, and possibly display an unusually shaped nucleus. As we can see in the animations, on the left side, this displays normal cell division. You can see conformed cell size and shape, an organized arrangement of cells. The cells possess appropriate structures for their function. They have a controlled level of division, and they do not expand beyond a single layer. On the other hand, the cancer cell division on the right side, you can see variation in cell size and shape, a disorganized arrangement, abnormal cell structures, an out-of-control rate of division, and it expands beyond a single layer. Third on the list would be samples of octopi tissue responsible for the complex ability to camouflage. These cephalopods have three different types of color-changing cells: chromatophores, iridophores, and leucophores. Chromatophores are pigment sac-containing organs attached to radial muscles. The radial muscle can dilate within milliseconds of a stimulus, giving octopi the incredible ability to change color according to their environment instantaneously. Iridophore cells, indicated by their name, reflect light as different colors depending on which angle is being observed. Think iridescent. Both chromatophores and iridophores are displayed in action in the top right animation. Leucophore cells scatter light, and when they have the full spectrum, the cells appear white, much how white hair from aging is actually transparent, not white. The root leuco, meaning white, may be familiar in the form of leukocytes, also known as our white blood cells. Last on the list would be samples from each level of toasted marshmallow. I'd like to use the power of a microscope to settle an important debate amongst my family. Incinerated is, in fact, not an acceptable flavor of roasted marshmallow. Bringing a raw marshmallow to the flame introduces heat, causing sugar molecules to oxidize, releasing hydrogen and oxygen. During this, a marshmallow is undergoing a beautiful chemical reaction known as caramelization. Too much heat, and the hydrogen and oxygen burn away from the sugar molecules, leaving only gross, solid black carbon. By collecting samples from each roast level, I can back a presentation with microscopic evidence that soot is no longer a marshmallow as we know it.